Perfect. All right, thank you everybody for being here. Today, as Sean just said, I'm going to talk about the observations I have made regarding the mating behavior of two different pseudoscorpions. So the reproductive behavior of pseudoscorpions have only been described for very few species. There's a huge gap here that needs to be filled. What we know is that pseudoscorpions reproduce by setting up a spermatophore in the soil or in the substrate that females will pick up later. And according to Weigel, there are three different ways in which this spermatophore can be picked up, can be laid by the male. So according to Weigel, there are those males that do not require courtship nor the presence of the female to lay the spermatophore. So they will be walking around, laying lots of spermatophores and kind of crossing their chela, hoping that a female would eventually pick them up. The other type of behavior that leads to the spermatophore deposition would be where males also don't require courtship, but they require to sense the presence of a female nearby to lay the spermatophore. And then the third type is those males that require courtships. Sorry. So that means that there's going to be a series of interactions between males and females that lead to the deposition of the spermatophore. And this occurs in many families, but there are the two families that I'll be talking about today, Carnetidae and Callipheridae, that are the families of the species that I have observed. Now, when we talk about the courtship behavior in Callipheridae, we need to refer to the Ramson organs, that are these two tubes that come out of the male genital operculum during mating, but we don't know yet what they do. There hasn't been descriptions of how they are used during mating. Like people have observed them, but really how they're used, what they're doing is still unknown. So I'll be referring to the Ramson organs for my second species, and I will be using the acronym RHO. Now, with my first species, I'll be talking about a species in the genus Lustrocernes. This is a carnetid that I collected in San Jose, Costa Rica, in the leaf litter of a secondary forest patch. And regarding this species, there have been, species in this genus, there have been two descriptions. One of them is for Lustrocernes argentinus that had been done by Palempete collaborators in Argentina. We still don't know the taxonomic identity of the species I'll be presenting here because because of the pandemic and other reasons, we haven't been able to send the specimens away to get a proper ID. We believe it can either be Argentinus or Silvestri just based on reports that have been done in the region. Now, that description that I'll be showing on here will be based on the observations that Palempete and collaborators did in 2019, in which they describe that the mating behavior of their studied species occurs in through phases, examination, spermatophore deposition, and sperm transfer that I also think fit the observations I made here. So for examination, males would approach the female. He will start with some initial chasing. Eventually, he would be able to approach the female, grab her chela, and he will constantly be grabbing her chela at the same time that she's kind of pushing back. He's trying to drag her around. She's also constantly touching the male. So this is what that behavior looks like. We see the male here. He grabs her chela. He's constantly touching her chela hands. She's also trying to grab him. He moves a lot along the space a lot, trying to grab her, but she's also kind of pushing back constantly. And this goes on for around 70 minutes. This leads to the second phase, which is the spermatophore deposition, in which they get into this position we see here. The male is grabbing the female chela with his own chela. Their mouth parts are very close to each other. It's almost as if they are kissing. And then they stay like that for a couple of minutes before he lifts his abdomen to lay the spermatophore. That is the one that we see here. So this is according to Weigel, the classic spermatophore for this species. We have a drop of fluid down here and the sperm storage here. And then the sperm transfer occurs. So male will walk back. He releases the female's hands. And if the female is receptive, she'll walk towards him with the ventral side of her body very close to the ground in order to pick up the spermatophore. So this is what we'll see here. He walks back. And then the receptive female walks forward. lifts up her body to pick up the spermatophore, and then he does this Kila wave with his hands. So I want to point out here that this is where my observations don't match exactly what Pali and Pietro and collaborators observed. My males do this kind of Kila wave at the end that they didn't observe at all in their observations. And in their cases, females will sometimes kind of aggressively react towards the male during this separation. I didn't observe that. So we can either point out that this is a different species whose behavior is being described, or if it's indeed Lustrocernus argentinus, it might be a population that is showing different behavioral patterns. However, I have very little observations. I need to get more observations in order to really see if this happens at all, like this aggressive interaction from the female. Now, for my second species, I refer is a dactylocalifer, Dactylocalifer sylvestris. It's found in the Midwest. I collected it in Nebraska, USA, under Yucca glauca plants in the prairie. The more closely related description I found was that for Dactylocalifer latrelli, which is found in Europe. It's a description from 1912. However, we now have more technology than we did back then. So I was able to get recordings that allowed me to do more detailed observations on how the Ramson organs are used during mating. So I'll be using this icon to represent this Midwestern species dance. I divided the mating behavior of this species into phases, what I call initial courtship, pre-insemination, insemination. It takes around 14 minutes for the whole process to go on. So initial courtship occurs this way. The male would approach a female, he would grab her hands with his own, and he starts walking around the place, always holding her hands, and he would constantly shake his body at the same time that his Ramson organs are being extended. So what we'll see here is a ventral view. So we are looking at the individuals from underneath, and this is what that behavior looks like. This is the male. He approaches the female. He still hasn't been able to grab her hands properly. I'll see if I can pause the video. Here, if you look here closely, you'll see the Ramson organs are being extended, and they go in and out very quickly. So they're out there and then in. And he's shaking his body as the Ramson organs are extended, and then he's moving along the space, Ramson organs are out, and always dragging the female with him. This goes on for around 6 minutes, and then this leads to the second stage, in which the male releases the female's hands, but they stay close to each other, facing each other. Now, this is a ventral view, a dorsal view, so we'll only see the Ramson organs as two lines here coming from the sides. And what he does is that he extends the Ramson organs, shakes his body, approaches the female, waves his kila in front of the female cephalothorax, and then he walks back and she walks towards him. And they will repeat this series of dance steps multiple times. So here is what that looks like. So the male will extend the Ramson organs there, shake his body as he approaches the female. He then waves his kila in front of her face, and then he walks back and she walks towards him. And they repeat the whole sequence all over again. And this goes on for around 2 minutes. And then they lay the spermatophore. This is the transition between what I call my second and third phases. So this is how the male lays the spermatophore. So we'll see here how the spermatophore is laid. This is another ventral view, so we are looking at them from underneath. And we'll see here where the spermatophore is being deposited. That's him laying the spermatophore right here. Look that the Ramson organs are fully extended during this time. And this is what the spermatophore looks like. This is not the greatest photo, but it's the best one I got. As he's laying the spermatophore, the Ramson organs remain extended. At some point, someone suggested that maybe they remain extended to guide the female towards the spermatophore. And notice here how he is using his first pair of legs to scratch the ground as the female gets close, and they get into this position that we see here. So from the dorsal view, the male is putting his cephalothorax underneath the females. His kila are grabbing the pedipalp coxal of the female, so that only her last two pair of legs remain on the ground. And that leads to the third and final stage of the mating, that is insemination. In this position, the male will extend his first pair of legs. His claws will drag along the spermatophore, anchor on the female's genital operculum, and then help her pick up the spermatophore. And this is what that looks like. He grabs the spermatophore, grabs her, and then helps her pick up the spermatophore. Now the Ramson organs are in and they don't go out anymore. And he starts shaking her. And this goes on for 8 minutes.
10 minutes.
Thank you. Now, as I said before, I'm interested on looking at the Ramson organs. So this is one of the preliminary things I've done. I was looking at how long the Ramson organs are extended along the mating. And we see here each of the colors represent one of the phases I just described for each of the mating pairs I had. So as a general pattern, the Ramson organs tend to be extended for longer as the mating progresses. And I compare only phase 1 and phase 2 because are the ones where there seems to be a lot of overlap. They are also extended more frequently during the first stage. And another observation that I was interested in is that on few occasions, the Ramson organs come in direct contact with the female body. At the same time, and also sometimes they will get in contact with the male's pedipalps that will then proceed to touch the female. So people have hypothesized that maybe they are using some volatile chemical cue, but with these observations, it also can point out to maybe some kind of tactile chemical cue that might be taking place. So in conclusion, for my first species, there are some differences compared to previous description, but I still have to make more observations to really see what this is all about, if it's just a species that is behaving differently in different populations, or if just the description for another, a new species that has not been described before. For Dactylocalifer sylvestris, my califerid, this might be, as far as I know, the first detailed observations of the use of the Ramson organs during mating. We still don't know what exactly their function is, but I am hoping to have an answer for this next year. So hopefully everything works fine and you can stay tuned for next year. And in general, I think this paper allows to contribute a little bit to fit a little bit of this huge gap we have on the mating behavior of this magnificent order of arachnids. And with this, I would like to thank you all for your attention, and particularly my lab, other advisors that have helped me figure out this project, people who has helped me, Sydney, who helped me a lot with the maintenance of the pseudoscorpions while I was doing this, and all of my funding sources, and I'll take any questions.
All right, that was awesome. And so far you don't have any questions. You just have everybody freaking out about how amazing those videos were. Oh, wait, there's a question. Does spermatophore quality decline with male age or number of mates?
I wish I had an answer for that. I don't have an answer for that. I don't know. We still don't know.
All right. Do females ever become aggressive towards courting males?
Yes, my first species, the females are very aggressive. Also, for the second species, I had a lot of rejections. Like I had 57 trials, only of which 7 made it. To be fair, I was also in the process of figuring out how to best record them. So I was like trying out different materials for the mating. So I don't know how much was just that it was not the right substrate or whatever. But females, if they don't want to mate, they will aggressively wave their chela towards a male. They won't let them get close to them. So I haven't seen them attack each other. But you can really see that the female will violently wave their chela in a way that males just stop approaching her at some point.
All right, so one more question. How readily do they deposit spermatophore stalks? Lots of stalks during every mating, or do they leave them around for females to find, or only during courtship?
So it depends on the species. More basal species will deposit spermatophores everywhere. But my study species would only do it after mating, and they have to go through all 3 stages to do it. There was never like a spermatophore laid without doing like the little kiss in the first species, or without going through like those dances that they do. So species, it seems to me that the species that require courtship require to go through all of it to do it. I did have a male who got rejected, and like he laid the spermatophore, and then the female didn't pick it up, and then they just laid the spermatophore there and they walked over it. But that was like the only rejection I got, aside from like those initial rejections where females just don't let them start at all.
All right. Well, thanks so much. I'm going to leave it there for questions, but I bet you if you attended one of these happy hours, people are going to be picking your brains about pseudoscorpions all night.
I'll be happy to talk about pseudoscorpions all night.
Great. Thanks.
Thank you.